Hello lovelies, in this video we're going to take you through vaccines for your GCSE biology. So we're going to talk to you about how they work, and then we're going to talk to you about how society feels about them as well.
Vaccines. First of all, let's look at how they work. So a small amount of dead or inactive pathogen with its antigens on are injected into the body. So these will not infect you and make you sick with that disease. They will or can cause an immune response, which we're going to talk about, which can give you things like feeling a bit hot, having a mild fever, maybe having some aches and pains. That might be how you feel after you've had an injection for a vaccine, but it's not going to be the pathogen that you've been injected with making you ill. The antigens in the vaccine are going to stimulate your lymphocytes. Remember, these are the white blood cells that produce antibodies, to make antibodies that are specific to that antigen. So they are going to be able to bind to it because their shapes will fit together. We call this the primary immune response. So your body reacting to an antigen by making antibodies for the first time. Some lymphocytes, remember, will also become memory cells. These are going to be able to produce these specific antibodies again much faster if you are actually infected with the real pathogen. So if your memory cells are activated by a real pathogen at some point in the future to make the antibodies, is what we call the secondary immune response. We need to be able to describe both the primary and the secondary immune response. So often it's depicted as a graph that looks like this, where we have the antibody concentration in the blood and then the amount of time along the bottom. So that initial point at the start of the graph is when you're first infected with a new antigen. So something that you've not been infected with before, a new pathogen that you haven't been sick with. Or if you've been vaccinated and you've been injected with that antigen for the first time. So this is then the primary immune response. Not many antibodies are made, so the graph line doesn't go up that high for this first half, because the response is quite slow, so it takes time for those antibodies to build up. That's because we have to go through the process of detecting the antigen and then making the antibodies and start replicating those cells to make more of them. That process is quite slow, and the antibodies don't actually remain in the blood for very long once they've been produced. So you can see we start to get a drop off and a fall down and a decrease in those antibodies. At this point, just before the second peak, that's when we're going to be reinfected with the same antigen. So this is important. It has to be the same antigen as was infected in the first part of the graph, and then we're going to get what we call the secondary immune response, because it'll be the second time we've come across this pathogen or the antigen in the body. So we get a much steeper, faster increase in antibody concentration, and the drop-off is much slower. So this is kind of the features of how you recognize the secondary immune response. This is because more antibodies are being produced much faster, because all we're doing is relying on those memory cells to rapidly start dividing, the memory B cells, rapidly start dividing, forming plasma cells, which are going to be producing lots of the antibody. More memory cells will also be produced, and these stay in the blood for longer and then are able to keep producing antibodies if needed by dividing to form plasma cells again. This means the immunity is slightly longer term and you get that slower drop off of the amount of antibodies in the blood. And like I said, the important thing here is that we only get that secondary immune response if you're reinfected with exactly the same antigen. Now, pathogens can be sneaky and they can change the shape of the antigens on their surface. So it's not always going to be necessarily a secondary immune response if you get infected with the same pathogen. It has to be the same antigen. And we'll look at antigenic variation next. Antigenic variation. Now, we mentioned this when we were talking about the primary and the secondary immune response. So it's really important that we understand how antigens can be very vital to what type of immune response the body is going to put out, and how pathogens have evolved alongside humans and our immune systems to be able to try and evade our immune systems. So memory B cells are able to produce a secondary immune response when they encounter a pathogen with exactly the same antigens that we have had inside our bodies before. This is because the antibodies that those memory cells make will be able to bind to that pathogen because they will match the shape of the antigen, and therefore they'll be able to remember they don't actually destroy the pathogen, but they will help to make sure that it gets destroyed by the immune system. Some pathogens are able to form different strains. So the pathogen is the same. For example, influenza virus is the same virus. It is still the influenza virus. It's not a different virus completely, but as a different strain because there'll be some minor genetic mutations which are able to change the shape of the antigens on the surface of that pathogen. This is called antigenic variation. And you can see here that I've got pathogen A, and then it's gone through antigenic mutation, and it's still pathogen A, but the antigen is now changed slightly. It no longer has that pointed shape. Instead, we've kind of chopped the top of that triangle off and we have a more flat shape. So if I was infected again with pathogen A after the mutation, although it's still the same pathogen, the antigens have changed. So this goes for vaccines as well. So if I was vaccinated with pathogen A, the antigens on pathogen A, or infected with pathogen A, and then I had a break, and then I caught or breathed in or inhaled through droplet infection pathogen A that had gone through the mutation, so the antigens were different, then the memory cells are going to produce an antibody for pathogen A with the pointy antigens, and they wouldn't work because they can't bind to the antigens on the new mutated pathogen because they're a different shape. So instead of having a secondary immune response to the new mutated pathogen A, we have to go back and do the primary immune response all over again. And this is evidence for how vaccines don't always work, and you need to get booster vaccines after a while, or you need to have a new flu vaccine, for example, every single year. And every year they mix together all of the most recent strains of the influenza virus in order to make sure the vaccine is the most up to date with the most up to date antigens we have, in order to make sure that the vaccine is effective. So this is something that we need to think about and be aware of when we're talking about the primary and secondary immune response. Herd immunity. This is a concept which explains how unvaccinated or vulnerable individuals, so some of those people on that list that we talked about, and how those people in a population can be protected from a pathogen due to a large proportion of the rest of the population being vaccinated and therefore being immune. Now, there is a certain threshold level of the number or percentage of the population that would need to be vaccinated for this to really work. And normally that's around 95% or more. There's actually a list where you can look up a disease or a pathogen and kind of what the percentage is that is sort of extrapolated from that, and what percentage you would need for us to have true herd immunity. So in the first diagram, you can see we have an unvaccinated population. That's all of the blue people. And then there's one red infected person there. If we then let this infection play out and spread through the population, you can see it spreads very quickly, rapidly, and it's spreading to all individuals because no one is vaccinated and therefore no one is immune. In the second diagram, again, we've got lots of blue unvaccinated people. We have a couple of red infected people, and then we have some green vaccinated individuals who will be immune. But if you look at the spread still occurs and goes quite rapidly to pretty much all of the unvaccinated people because there's a low proportion or below the threshold percentage of people in the population who are vaccinated. In my final diagram, though, you can see we have a much higher percentage of vaccinated people and immune people. And so although we have about 4 or 5, so we have double the number of sick, infected individuals, the red individuals, what actually happens when we let that virus spread is it can only really spread to a few more individuals nearby to those people who are unvaccinated. So that means the majority of those unvaccinated people in that population are protected and they don't actually get the virus. This is because if there's enough immune people in the population and they're immune because they've been vaccinated, then there's fewer pathways for the virus or the pathogen, whatever it is, to spread from one individual to another. And so it gets stuck, it stops, and it can't spread to a bunch of people who are all immune, and they can't pass it on to anyone else. And so therefore the chain of infection is stopped. And that means that the majority of those unvaccinated in the population, who should be the people who cannot be vaccinated for medical reasons or age reasons or because they have that weakened immune system, they are protected so they will not get sick even though they can't be vaccinated. So if you have to describe or explain herd immunity, describing it is by saying that it's when a large number of the population are vaccinated so that they are immune to a certain pathogen or disease. And then the way it works is that it means that the virus is then not able to spread from person to person because people who are immune cannot carry or pass the disease on and spread it to other people. And the reason this is useful is that it protects the people who cannot be vaccinated. So recently, there's been a decrease in the number of children getting vaccinated for various diseases, so viral diseases, including serious ones like measles in certain countries. This has actually created a large increase in the number of cases and even deaths from viral diseases that would normally be incredibly rare because of a high amount of herd immunity that existed when everyone was getting vaccinated up to this point. So it's important evidence for the fact that herd immunity works, and having a high proportion of the population vaccinated against diseases can actually reduce their spread and reduce their chances of being able to spread through the population. It's also how we actually eradicated some diseases like smallpox, for example, which doesn't exist anymore. And that's because we managed to vaccinate so many people against smallpox that there was no host that the virus could live in anymore. So it couldn't replicate, it couldn't pass from person to person. And when the last person with smallpox died, it wasn't able to spread to anybody else. And so we actually don't have that virus spreading around in the population anymore.
Ouch! This is why in some videos I'll explain scratches.